Thank you, David. It's a real pleasure to be here and to be able to talk to you tonight about cancer of the brain and cancer and the brain. And so I've actually divided this talk into two parts. The part on cancer of the brain really is the sort of thing that I've been working on for the last couple of decades, trying to deploy genomic analysis technologies to tell us how cancers of all types work. You know, what are the fundamental parts of the cancers that go awry and lead to the formation and progression of the cancer. And one of the cancers that we've focused on in this national initiative that I'll tell you about is brain cancer, in particular glioblastoma. And so I'll spend the first half of this talk telling you a bit about that. The second half of the talk is going to be something that is actually new to me, and it derives from our increasing understanding that as we try to treat cancers, mostly we focus on the cancer, but one of the things that we are increasingly appreciating is the concept that it's not just the cancer that we need to be treating. The cancer resides in a very complex milieu in the body. And it's actually very important what parts of that milieu the cancer cells sit in, because they influence how the cells are going to behave and how they're going to respond to treatment. And one of the interesting things to me is that the brain plays a very important role in adjusting the body and how the cancer grows in it. And so the second half of this talk is really going to be my sharing with you some of the things I've learned about how it is that the brain can be impacting cancer. And I think you will appreciate that as we learn more about the brain and cancer, one of the things that we're going to have to learn to do is to manage the brain as well as the cancer when we try to treat these diseases. So I'll take you through both of these stories today. And before I do that, I am required by OHSU to inform you that I have several industrial connections that might be viewed as conflicts of interest. I'll let you read the list here, but the only one that is vaguely relevant to today's talk is the fact that I have licensed some intellectual property that I'll mention in passing today to Abbott Diagnostics. So beware of that. So, moving on to the topics for discussion today. The first thing that I'm going to talk about is cancer of the brain. And this is intended to give you an understanding of how it is or how much we've learned about cancer of the brain in the last little while. I'm going to start the discussion by telling you a little bit of history, and in fact I'm going to retell a bit of the Brian Druker story because that is in fact the motivating force behind a lot of the work that we're now doing trying to understand cancers of all type. So I'll tell you a bit of history there. And then I'm going to talk specifically about brain tumors, and in particular, a particularly aggressive form of brain cancer called glioblastoma. And I'm going to talk a little bit about what we've learned in the last few years about both its genesis and its progression, and to give you an idea of how we're using this information to devise new therapeutic strategies. So I'll start with that. And then I'm going to talk, as I mentioned, about cancer and the brain. And I'm going to talk about two particular things. One of them is I'm going to talk about the concept of stress in cancer, because it turns out that stress influences the brain. That signals to cancers around the body and influences how they behave. In the second part of the talk, I'm going to talk about cancer-associated wasting, cachexia in some jargon. And this derives from signals that the brain gets from the cancer and misinterprets them in ways that lead to this concept of wasting. So I think both of these things are fascinating stories, and I'll tell you a little bit about some of the work that's going on in those areas. So let me start with the part of the talk on cancer of the brain and cancer as a disease of the genes. And just so we're all on the same page, what goes awry in cancer, brain or any other thing? It's basically abnormalities that form in the DNA that encodes the molecular machines of life. So basically DNA is a molecule. Every cell in the body has DNA. If you were to stretch out the DNA in any cell, it would be about 6 feet long. And it turns out that this material is more or less an alphabet that encodes the words that can be interpreted to allow cells to make the proteins that comprise them. And then the proteins are organized into molecular machines. The molecular machines are assembled into cells. The cells are assembled into tissues. And when you get cancer, what you've done is to corrupt the DNA in the cell in ways that it messes up the proteins, cells, and tissues in ways that allow cells to become acivil, to invade throughout the body, and ultimately to perhaps kill the patient. So that's the process that we're trying to understand. And in order to do that, we have to understand what goes on at the DNA level. And so, as I mentioned, every cell in the body, and there are about 10 trillion of them, is instructed by the DNA molecules that are encoded in about 6 billion letters of DNA. And these things are organized into the bodies called chromosomes. So there's 24 pairs, 48 different chromosomes. You see the chromosomes as you would view them in a microscope. If you looked at those from a single cell. And on the right, you can see one of those chromosomes exploded so that you can see the individual DNA molecules. And so about 20 years ago, the challenge was that's pretty much all we knew about DNA. We knew there was about 6 meters of it, but we didn't know what the code was. And one of the major, in my mind, one of the major scientific accomplishments of the last century was that the scientific community worldwide got together to decode the mostly complete DNA sequence of a normal human. And this was actually something that is very remarkable in the field of biology. It's probably the first time that there's been such a large international effort to accomplish something of this magnitude. It was an international effort. It involved both the public institutions in the countries and it involved the private sector. And in the end, there were literally thousands of scientists working together to try to figure out how to do this, and they both worked together and they competed. If you were around during those days, the Time magazine cover here indicates that this was actually a competition between the public sector and the private sector, with Francis Collins leading the public sector and Craig Venter leading the private sector. It actually spurred us right along. And in the end, got us to the point where in about 10 years, we had gone from having this more or less featureless mass of spaghetti to knowing pretty much what its complete sequence was. Took 1,000 people. Cost about $1 billion, give or take. And so that, another remarkable thing about that particular story is that unlike most activities in the public and private sector, the data was made available to the community as it was generated and interpreted. So that was another sort of landmark change in how science was done in this country, is that the data was available to anybody who wanted to use it. And today, you can access the whole thing on the internet if you have the right URL. So that's pretty remarkable. We know what all the genes are. We know what many of the regulatory sequences are. We really, however, are just beginning to interpret how it is the DNA actually works. But we have some clues, and some of those clues have turned out to be quite useful in the management of diseases, in particular cancer. So, as I mentioned before, cancer is, in essence, a disease of genes gone bad. This happens through a variety of mechanisms in life. Your cells have to replicate DNA as they divide and grow, and sometimes that process isn't perfect. And sometimes you experience chemicals or radiation from the environment that breaks DNA. There are many mechanisms by which DNA can be damaged, but in some cases, it gets damaged in a way that allows cancer to form. And the prototypic example of how this happens was really illustrated by Janet Rowley almost 40 years ago now. In 1973, she was studying then a reasonably obscure disease, chronic myelogenous leukemia. And one of the things that she discovered by looking in the microscope at these things called chromosomes is that in a large fraction of cancer cells from patients with CML, there was a specific rearrangement between two of the chromosomes, chromosome number 9 and chromosome number 22. And this rearrangement actually formed a small, then called and still called, Philadelphia chromosome, a new chromosome that brought two genes together. And this appeared to be causal. And so there was a lot of excitement about this in terms of being able to diagnose and characterize the disease. I entered the scene about that time, and working with colleague Dan Pinkel, actually invented some ways that help us to diagnose that disease by, in essence, developing some chemistry that allows us to paint particular parts of the genome different colors. And what this allowed us to do was to easily identify patients that carried this genomic abnormality. And that was important because one of the things that we wanted to be able to do was to target those patients with new therapies. Enter Brian Drucker. So Brian was studying CML at that time, and in particular, he was studying the gene, this new gene fusion that was caused by bringing these two bits of the chromosomes together. The two genes are called BCR and ABL. And one of the things that Brian discovered was that the fusion of these two genes turned on a process that led to the proliferation of CML in ways that allowed those cells to grow unchecked. And more importantly than that, working with now a company called Novartis, developed a drug called Gleevec that allowed us to block, or allowed him to block, the function, this activating function of this BCR-ABL complex in ways that prevented the cancer from progressing. And so this was really the first example where people had learned to take information about a genomic abnormality, understand what the genes did, and then do something about it chemically that led to the strong control of patients with CML. It really took the disease from being a death sentence to being something that is reasonably well managed in the long term today. So that was exciting, and it caused the rest of the world in general, and Brian in particular, to want to do this again. And that is the genesis of the new campaign that we have now launched, the so-called One Down, one cancer down, many to go. And so Brian is now attempting to help us here in Oregon do the same thing for many other cancers. And so one of those might be brain cancer. And so that takes me to the second part of the talk that I wanted to give, and that is glioblastoma. This is a very aggressive cancer, as I will tell you about. And so one of the things that we need to know about this cancer is what are the genomic changes in the disease that lead to its formation and progression and response or not to treatment. So can we do this same thing for the brain? And we're getting some help in this regard by another national project that was formed at the conclusion of the effort to sequence the Human Genome Program. At the end of that, several of us got together with Francis Collins, who was then head of the institute at the National Cancer Institute that did genomic research, and the leadership from the National Cancer Institute, and we said, can't we accelerate this process a bit? Can't we band together as a community and use this same team spirit that we had in sequencing the normal genome in the first place to sequence cancers. And so the Cancer Genome Atlas Project was initiated in 2006 with that as a goal. Basically, the goal of the TCGA project, as it's now known, was to find targetable aberrations in essentially all of the major cancer types. So instead of spending all of our time determining one sequence, this project has its goal determining the genome sequences of about 25,000 cancers. And so that was an ambitious undertaking, but what we're looking for are things that corrupt the DNA, things called mutations that change the code itself, things that are called translocations. That's what Janet Rowley found in CML, where you rearrange parts of the genome, changes in genome copy number. You have too many copies of the genome in one place, too few copies in another, or what we call epigenomic modification, which is just putting little chemical modifications on the DNA that cause it to be processed differently. And so Paul Spellman, who I'm illustrating here, was recently recruited along with me to OHSU. Paul is a leader in the TCGA project and is sort of our link into this particular enterprise. The question, the first question was, is there anything there to find? It's really a discovery process. And so again, my colleagues and I now in California, or then in California, actually invented a couple of techniques that allowed us to take a quick view into these cancers and to ask the question, is there anything there to be discovered? And the answer turned out to be, unfortunately, a lot more than we had initially imagined was going wrong in these cancers. So these are genome maps, if you will. This is the entire stretch of DNA in a normal individual. And what I'm showing you here, just in this little plot, are changes from normal. Anytime you see something go up, that means there's an abnormality there that makes too many copies of the genome. Anytime you see a blop go down, that means that cancer has lost that part of the genome. And the interesting thing about it is that there's not one or two abnormalities that are present in these cancers, and this is true for almost any cancer. There are dozens to hundreds, and so many of these things we know enough about to think of as therapeutic targets. So what we're trying to do in the TCGA project in the context of all of these different cancers is to make maps of these abnormalities so we can find the abnormal genes and target them therapeutically. Well, there was a lot of debate about what cancer to tackle first when this project was first initiated, and glioblastoma, brain cancer, was chosen as the first of the things that we were going to go after because of the fact that this is the most common and the most lethal form of brain tumor. And I'm just showing you some statistics here to make the point that this is a really very poor outcome disease. You can see three different classes of cancer depending on how they present clinically and respond to surgery. But basically, the historical median survival time for cancers ranges for the good ones from like 17 months down to 7.5 months. That's the median survival time. If you look at the 5-year survival, sometimes as many as 14% of patients in this class will survive that long, but if you're in this class, there's almost no chance of surviving that long. And so the TCGA decided, because of the really poor performance of this tumor, this was where we had to start. And so we started the process in 2006, and by 2008, we had pretty much completed the first map of glioblastoma. And again, this was a pretty remarkable scientific collaboration. This was done by 200 scientists in 60 different academic institutions, all collaborating together on a weekly basis to try to invent the technologies that were needed to map all of these genes, and then putting the information together in ways that could be somehow interpreted clinically. Well, another interesting thing about this is the way the publication was cited, and you'll notice that there aren't any authors listed. It's actually listed under the authorship of the consortium. So this particular project has actually changed in many ways how this part of the scientific community works together. The goal is to get to the answer that we're looking for medically rather than to advertise any particular individual's contribution to it. So I think this has had this project has had a fairly major sociological impact on the whole scientific community. Well, what have we learned about glioblastoma? And I'm going to give you a bunch of letters, but it's pretty interesting. The first thing that we've learned is that there are some frequently mutated genes in glioblastoma. Here's a list of them. We know a fair amount about most of these things, and I'm going to tell you a story about one of them. I'm going to tell you a story about this gene. Turns out that it's known to be involved in the metabolic activity of glioblastoma. We know a lot about the other ones. I'm only going to talk about this one, but there could be comparable stories told for almost any one of these genes. And I'll tell you what now the scientific community is doing with the information that this gene is frequently mutated in glioblastoma. Well, the first thing that we've used is to understand how the patients that have this abnormality perform clinically. Now, I'll show a few of these curves during the evening, so let me tell you what it's telling you. This is the fraction of patients that were present in this clinical study, and this is how long they survived. So as this curve goes down, you'll see that there are fewer and fewer patients that have survived this length of time. So about 20% of the patients survived 30 months, and about 10% of the patients survived 60 months. This is if they had a normal copy of the gene. But one of the things that was interesting about the patients that had this mutant is that they did considerably better. And so that said, yes, this gene is probably important in the disease, and it is somehow influencing the way that the cancers behave. We recently had a visitor from Andrew Trister, who's in the radiation oncology program at the University of Washington. He's been studying this disease using a brain imaging technology. And one of the things that he's discovered is that, in fact, the way that these cancers invade into the brain is very different depending on whether or not you have the mutation or whether you do not have the mutation. And for whatever reason, the tumors that have the mutation seem to be more invasive than those that are not. That's a little counterintuitive, but that's the biology that we're now trying to understand. Well, what do we do with that bit of information? That's biologically interesting, but I think from a therapeutic point of view, one of the things that this has allowed us to do is to say, can we find chemical inhibitors of the protein that's encoded by that gene? And so the answer to that has turned out to be yes. There's this obscurely named compound up there that, in fact, when given to cancer cells that carry this mutation, turns out to be effective in terms of inhibiting the way that they grow. Now, if you remember the story that I told you in the beginning of going from a discovery of a translocation to the drug Gleevec for chronic myelogenous leukemia, that took about 30 years to play out. One of the nice things about the TCGA project is that the public is paying attention to the data. The data are available, as I mentioned, as they are discovered. And so it has now taken us only 5 years to go from the discovery of the mutation to the development of a new experimental drug. And so I think that's exciting. It's really accelerating the pace of progress in the field in a very beneficial way. And as I say, there are other genes in glioblastoma that are equally attackable, and the same sort of thing is going on elsewhere. So that's one of the things that we've learned from the project. Now, the other thing that we've learned from the project is a bit about how brain tumors evolve and about why it's difficult to treat them. So this is a little obscure in presentation, but essentially this is a story from my colleague at the University of California, San Francisco, Joe Costello, who was studying brain tumors as they progressed. And it was taking biopsies at the time of presentation, the first recurrence, at the second recurrence, at the third recurrence, and applying these same genomic analysis technologies to each of these tumors. And one of the things that he discovered was that the recurring tumors look very little like the primary tumor from which they were derived. They have evolved an amazing number of new mutations that are just not present in the primary tumor. And so if we were trying to base our treatment of the recurrent tumors on what we knew about the primary, we'd probably not be doing it right. So these tools now give us the ability to follow the evolution of these tumors and to follow how it is that they change genomically. The other thing that the community discovered was that some of the drugs that we use to treat these things have the effect of altering the way genes involved in DNA repair are expressed. In essence, it turns them down. And so one of the things that this particular drug seems to do is to turn down a gene involved in DNA repair that leads them to become hypermutatable. The mutation rate goes up dramatically when you knock down this gene. And that's one of the reasons why these tumors are evolving so rapidly, is we've turned off an important DNA repair machinery. And so that's a really important bit of insight that tells us, well, maybe we need to think differently about how it is that we use this particular therapeutic compound. So it's helping us to understand how it is that these tumors are behaving, how they're evolving genomically. And so that's turning out to be, I think, something that's going to be routinely useful clinically. And in fact, we are anticipating that this kind of an analysis is going to become part of the standard of care, not just for brain tumors, but for essentially all cancers in the near future. And one of the things that makes that possible is the fact that from the time that I stopped working on technology to analyze tumors till today, the genome analysis price, speed, performance has increased over 1 billion-fold. That's pretty, I mean, it's very hard to say that about anything else that we've done in the world, but we have improved things about 1 billion-fold. And it's taken us from the $1 billion genome to the $1,000 genome, and it's taken us from something that took about 10 years to complete to something that can be done in a week or so. And so we're imagining that we're going to be generating millions of genome sequences, and we're going to have to interpret all of this. So this is an amount of data that's actually pretty hard to imagine. I recently tried to compare it to something that I knew about, and if you think about how much information is encoded in the Library of Congress, I'm imagining that in a few years we will be processing on an annual basis about 10,000 times that amount of information. So that's a pretty mind-boggling thing to do. And so one of the things that's really exciting about the Portland area is the presence of Intel. And so one of the things that we've now done is to form a partnership with Intel to build a new generation of computers, actually a computing cloud that we think is going to allow us in a very inefficient way to manage and interpret the data that's out there. And so we've now set up a formative collaboration between Intel, OHSU, Memorial Sloan Kettering in New York, the MD Anderson Cancer Center in Houston, with the idea of building a prototype of this system in ways that we hope will allow us to acquire experimental data and ultimately interpret it in ways that will be clinically useful. So that takes me to the end of the first half of this talk. I think that international genome aberration discovery efforts have really defined the genomic landscape of glioblastoma and other cancers. This information is really now already leading to the development of new diagnostic and therapeutic approaches that I think are pretty exciting. I think that it has taught us that we still have a long way to go. It's shown us how rapidly these tumors can evolve and the fact that we're going to have to pay attention to them as they do evolve if we're going to continue to develop durable therapies. But that's sort of a daunting message. And so that realization, this rapid evolution of advanced cancers is one of the things that's really spurred Brian Drucker and those of us in the Knight Cancer Institute to try in this new project to increase emphasis on earlier detection of these lethal cancers when they have lower genomic complexity. And so that's, as I say, one of the founding principles of the One Down program that we are now initiating. So that's genomics. Now let's talk about cancer and the brain. And I want to make the case now that what I'm about to tell you about is that the brain was involved to maintain homeostatic behavior in humans. It turns out that the brain is a remarkable organ that receives information psychogenically, just interacting with the world, and physiological. It receives signals, chemical signals from throughout the body, especially the immune system, that pretty much inform the brain as to what's going on. And the brain takes this information, and a particular part of it then interprets it in ways that sends signals back out to the body that influence a lot of what we do. These signals can influence metabolism, they can influence the cardiovascular state, they can influence cognition, they can influence immunologic function, and so on. And the whole goal of this process is to restore the homeostatic state in humans in response to changes from normal. Well, one of the changes from normal is the induction of stress. And there are really two kinds of stress. One is acute stress. This is probably escaping from the saber-toothed tiger, but this is the fight or flight response, and the body needs to do something and do it quick. And so I think that many of the response mechanisms were in fact evolved to do this. What I'm going to tell you about is that some of these same mechanisms, when exposed to chronic stress, actually lead to some pathologic conditions. The story I'm telling you is one that I've been learning from my colleague Anil Sood at the MD Anderson Cancer Center. We've collaborated over the years on a variety of areas, and I've been fascinated by his elucidation of what's going on. What are some of the mechanisms that actually are going on here? Well, how does the body, the brain, react to an acute stressor? Well, you need to run or fight, so you need to turn up a metabolism of energy, and you inhibit further storage of energy. You increase the heart rate, blood pressure, breathing, perception of pain is blunted, cognition is sharper. The brain is taking care of all of these things by manipulating the systems that I just told you about. Well, what happens if you have chronic stress? Life in the real world. Well, this turns out to have a number of untoward effects. Basically, physiological, it can generate depression, anxiety, lead to heart disease, strokes, susceptibility to infection. I'm going to talk about its impact on cancer progression, but there are other things: GI problems, diabetes, pain, sleep disturbances, and so on. Well, how might stress influence cancer behavior? And one of the things that we've come to learn is that the behavior of every cell in a cancer is influenced by signals from the environments in which that cancer resides. They reside in very many different environments, and so the concept here, in terms of the stress in the brain idea in cancer, is that stress-related signals from the brain actually influence some of the environments in which cancer cells reside, and they influence the cancer cells themselves. They have impact on cells that are surrounding the cancers, they have impact on blood vessel formation, they have impact on the immune system, and so on. And essentially what we're dealing with when we're trying to deal with cancer is the fact that you may have a cancer cell that's trying to grow, to die, to differentiate, progress, respond to therapy, and so on. And all of these responses are modulated by the signals that it gets from the environment. Well, we actually know how to study this a little bit just to see if there's any truth to this hypothesis. And so one of the things that we've taken to doing in my lab is to make what we call microenvironment microarrays. We can take proteins from all over the body, make little spots of them, Each spot has a different protein or combination of proteins. And then we can take our cancer cells and put them on top of those environments and ask what they do. And what they do is a lot. It turns out that we can find a number of environments that will cause the cancer cells to go completely dormant. We can find other environments, same cancer cells, where the cells grow incredibly rapidly. We can find environments where the cells respond vigorously to our therapeutic agents. We can find environments where the cells don't respond at all. So all of these environments influence dramatically how it is that these cancer cells behave. Well, what does this have to do with the brain? So back to this signaling process that I told you about before. The brain, in response to stress, in response to acute stress, generates some of these signals that influence immune cells. The signals influence blood vessel growth. They influence surrounding cells and the tumor cells themselves. And so what do these signals from the brain actually do? They cause the immune system to be depressed in some cases. They cause the cells to become, the cancer cells to become more able to migrate and invade. They alter the ability to repair DNA. They decrease cell death, which is not what you want to have happening in your cancer. It increases new blood vessel formation so that the cancers can continue to progress. And they even influence the ability of viruses that are associated with cancer to replicate. So this is what biology would tell us should influence the way that cancers respond to stress. So what's the experimental evidence that says that this is true? Well, one bit of experimental evidence comes from the study of mice that carry cancer cells. And the idea here is that you can take these experimental animals and expose them to various forms of stress, for example, restraining the mouse a little bit. And what this does in this experimental model is to show very clearly that induction of chronic stress, just as we were talking about, promotes tumor growth and blood vessel formation, in this particular case, in an ovarian cancer. And there are a lot of such studies, and the answer generally seems to be the same. If you put animals that are prone to develop tumors under stress, you generally increase metastasis, the invasion, the mobility of cancer cells, increased growth rate, increased metastasis again. And if you inhibit some of these processes, you can actually decrease these processes. So inhibitors can decrease growth and decrease cancer formation. So it looks like, at least in animal model systems, that this concept that the brain can directly influence the way that cancers behave is true. Well, is there any clinical evidence for this? And it turns out in this particular study here, they went through the literature and documented the fact that there are 330 published studies that suggest that these stress-related factors were associated with poorer overall cancer survival. Now, these are not huge effects. These are subtle effects. So some of the strongest effects are in liver cancer, head and neck cancer, and so on. You know, it improves or is detrimental by a factor of about 2. 53 studies, stress-related factors were associated with higher cancer mortality. So it does seem that, in fact, stress overall does play a negative role on cancer outcome. Well, can you do anything about it? And there is an emerging literature that suggests that the answer to that may be yes. So here's one study where they were using beta blockers to improve relapse-free survival in patients with triple-negative breast cancer. There are several such papers. If you do a summary of the effects of beta blockers on cancer-related mortality, the longer the bar, the bigger the effect. These are a whole bunch of different cancer types. And in general, what you can see is that beta blockers in this particular analysis appeared to be beneficial in quite a reasonably large number of cancers. So to me, as somebody who is trying to come up with new strategies to think about how it is that we should manage the treatment of cancers that I work on, which are primarily breast cancer and pancreatic cancer, this has now caused me to think that we need to bring the brain into the equation and to make managing the brain as much a part of treatment as managing the cancer itself. Well, the last story I'm going to tell you is one about cancer-associated wasting. And this is a story of signals from the cancer to the brain and the brain reacting inappropriately to those signals. So I think you all know this story. This is the Patrick Swayze story where pancreatic cancer carried him from this robust physical specimen to a person who weighed 105 pounds shortly before his death. And I'm going to try to make the case in this part of the talk that the influence of the cancer on the brain is the thing that's actually driven most of this. Now, if you read the internet, you will find a number of articles that attribute this particular wasting to chemotherapy. And there's no doubt that there is some truth to that. I mean, that's a slight part of the problem. But I think that I'm going to make the case now that, in fact, most of what causes this wasting is inappropriate signaling to and from the brain. Now, this is what I'm going to talk about is not my research, but it's going to be research from the laboratory of Dan Marks, one of the world experts in this particular area and a OHSU faculty member. This is Dan and his laboratory. For those of you who care about this particular part of it, Dan has written this article in Physiology and Behavior about this particular process that I highly recommend as something that you might want to take a look at. And when Dan starts these talks, he basically talks about sickness behavior. And so what happens when you get sick? Well, first off, why do you get sick? You get sick probably because you have some sort of an infection. And what the body does is you aren't hungry, you get tired, don't sleep well, you get depressed, unrestful, increased sensitivity to pain. And essentially, what Dan argues here is that this is an adaptive response to infection. And so what the body is trying to do is to minimize energy expenditure and to divert resources to fight infection. And so what happens then? Well, you increase the basal metabolic rate, you sort of digest proteins in muscle to provide energy, you change the immune system to turn it up and generate a fever and so on. Why do you generate a fever? Well, turns out that fever generates a body temperature that favors the proliferation of immune cells, but is hostile to pathogen growth. But it's expensive. It takes a lot of energy to increase temperature, and you do a lot of things to try to increase energy in general. So, okay, what do you need to do? Well, in order to get this energy, the thing you have to do is to digest your muscles. This is accomplished by breaking the muscles down to release energy and protein building blocks to mount the immune response. And it's not a very efficient process. So in order to mount a proper immune response, you end up having to break down a lot of lean muscle. I'll come back to that. This is the part of the brain that does it. I'm not going to go into the details. There'll be a test later on this. So here's the process. If I have an infection, the local immune cells are going to recognize the presence of the pathogen. And one of the things that they're going to do is to send out signals, cytokines. These are the names of some of them. These signals get picked up by the brain, and the brain then amplifies the signal. It says, okay, there's an infection someplace in the body. Let me help the body get ready for that. So one of the things that it does is it starts altering these behavioral and metabolic outputs, and it sends signals to the muscles that I just talked about that says, okay, let's make some energy for protein synthesis. And so this then mounts the immune system or immune response that you need to get rid of the pathogen. Well, the important thing about this process is that this whole cycle ends when the pathogen is eliminated. Well, unfortunately, in cancer, you get the same sort of signals going to the brain, but unless you've eliminated the cancer, they continue to go to the brain. So now what you get is a long-term alteration in behavior, metabolic outputs, and you tend to continue to degrade the muscle to provide the immune system with support. And this is unfortunate because if it never ends, this leads to pathological conditions. It leads to this muscle wasting. And in many diseases like pancreatic cancer, it's the wasting of the body that's as important in the demise of the patient as the cancer itself. So what would happen if you could actually prevent this? And there are some studies that are now beginning to emerge that suggest that being able to prevent this process, to stop that feedback from the brain, could be enormously beneficial. So back to a mouse experiment for a minute. This is an experimental tumor-bearing mouse that experiences this muscle wasting. And it turns out that this particular group has developed a prototypic antagonist of the muscle wasting machinery. And so if you treat the mice with this antagonist, you reverse the loss of muscle, and the mice survive. If you don't, the mice rapidly progress to death. I'm not going to walk you through all of the details here, but suffice it to say that when you treat with this antagonist, body weight goes up, and the mice that have cancer survive substantially longer than that. The Marks Laboratory is exploring this in great mechanistic detail. And I'm not going to go through the biochemistry of melanocortin-4 receptor antagonist, but that's what they study. And one of the things that has become apparent is that the signals, these inappropriate signals from the body going through the brain and in this amplification cycle require this particular receptor. And what he's determined is that it's now possible to block that particular receptor. And in doing so, what it tends to do is it improves energy, it reduces loss of body mass, and generally improves the psychological conditions of the patients. So this is very promising. This is still very much a work in progress, but the Marks Laboratory is, I say, one of the world's leaders in this, and I think that this is another aspect of brain chemistry that we're going to have to learn to incorporate into our cancer treatment strategies in order to have the most robust possible therapeutic approaches. So just to summarize all of this, I think that both acute and chronic stress can adversely influence cancer progression in response to therapy. I think that clinical management of stress can actually improve outcome in some cases, and we need to think about that in the psychosocial management of cancer. It's very clear that the brain responds to cancer-associated chemical signals by activating these mechanisms that actually have evolved to fight inflammation. In the long term, it turns out that if you have cancer around, they lead to wasting and other adverse events. But I think that Dan and other people in the community are now beginning to make real progress in understanding the mechanisms and that really suggest new strategies to manage this process. So I think it's a special time in cancer. I think that we know a lot about cancer of the brain. I think that we have really great clues now in glioblastoma and other cancers of ways to attack the aberrant genes that are present in it. And I think that we are now beginning to appreciate much more than we have until very recently the importance both of stress and cancer-associated wasting as diseases of the brain that can be managed to a therapeutic benefit in the patient. And so that's the end of the story I wanted to tell you. I want to give a particular thanks to Dan Marks and Anil Sood, who provided me with some of the education and the slides that I used in this presentation. Thank you.